Yeah. So today is today is one of the regular meetings for the ICAN network, which basically is the network which links both Singapore, Denmark, and also the US together in the context of cellular agriculture. So we set out to actually try to exchange some of our ideas in relation to research related to cellular agriculture, and we do have actually other working groups under this network. So for those people that has dialed in to learn about what Alcine and also Nicola is doing, if you're interested, you can come and talk to me. For those in Singapore, I can actually share a bit more. And if you're interested to visit or work in different places, whether it's the US or in Denmark, we can also link you up, because I think this is what one of the network aim is, to exchange some of these people and ideas and so forth. So it goes beyond just exchanging research ideas and so on, and also actually exchanging people from different places. So without further delay, I will invite Nicola to share with us what DTI has been doing. I will also invite Nicola to also give yourself a brief introduction as well before you start. Yeah.
Okay, so my name is Nikola and I'm working at DTI. I did a PhD in microalgae, just basic ecophysiology of microalgae, and then after the PhD I got a position here at DTI where I'm working on microalgae and their application in biotechnology. So I'm coming from a biology background. And now I will give a presentation. Okay, I need to send a request for sharing the screen, so you need to accept it.
Okay. Does it work now?
Can you see now the presentation?
Yep. Yes.
Is it in presenting mode? Yes.
It's in your speaker mode.
Uh,
You might want to go up to the display settings, I think.
this one, display setting, and this one, swap presenter. This one. Now
Yeah.
you can see. Okay,
Yeah.
perfect. So I will give a short presentation about the Danish Technological Institute, which is a self-owned non-profit institution, and it's a research and technology organization which kind of makes a bridge between academic research towards us and then to different type of companies. We are covering all aspects of technologies, and we have more than 80 laboratories with all the equipment on the state of the art, and also we have a central facility next to Copenhagen, and then we have a few other facilities around in Denmark, and also we have one office in Spain. The institute is divided in 5 divisions, which is environmental technology, energy and climate, materials, building and construction, and food and production. And this is also where the Center for Bioresources, where I'm working, belongs. And we are a small team of 3 of us working in the Center for Bioresources, and we are working on microalgae. We're working on different aspects of microalgae, and we're also recognizing the ecological importance of microalgae, because microalgae, they're important as producing oxygen, and they're living in all aquatic environment, which ocean is the biggest ecosystem on Earth. Then they're also supporting the biggest food web, which is the oceanic food web. And also they're showing when they're indicators of the ecosystem imbalance. And also we all know that they absorb most of the CO2 from the atmosphere. But of course, we also know that we can apply microalgae in different areas. So they have a huge economic importance in biofuel production, food production, and aquaculture and feed production. So we are considering microalgae as a part of the biosolution, and also they have an important role in the green transition, where we are moving from fossil economy to sustainable economy. And we can use actually microalgae for different crises that are now going on in the world, which is the climate crisis, biodiversity, environmental, protein crisis, energy, and water crisis. And we can produce biomaterials, sustainable cosmetic, clean energy, and also sustainable food and feed through microalgae. And of course, we can contribute to biodiversity with clean products. At DTI, we have a facility where we can cultivate microalgae from single cell, which is at lab scale. We can upscale the cultivation of microalgae in the indoor photobioreactors, and then also we have the outdoors photobioreactors. And for now, we are working on different aspects of microalgae biotechnology, and also we are part of a lot into R&D projects. So now I will try for each topic to present some of the projects. So the first one is in microalgae and feed. And this was one project which was called REMAP, where we were using resource-efficient microalgae protein production. And this is how it looks in practice. So there was a biogas plant, there was a CO2 supply and a sunlight. And this was an outdoor cultivation system where microalgae were cultivated outdoors. Then there was a harvesting and all the downstream processing of the microalgae, and then the formulation of the proteins they were using for feeding trials in chickens. And of course, the food safety and analysis was done. And why we choose microalgae? Because microalgae, they contain actually methionine, is the sulfur-containing essential amino acid, which is lacking, for example, in the soy-based chicken feed. And also you can cultivate the biomass productivity ton per year was 26.7, while for soy it's 2.5, if you compare soy and microalgae. And also for microalgae, actually, you can cultivate them on non-arable land and they don't require pesticides as crops requires. And then another project was also called protein from locally grown legumes and algae for organic chickens. And this was again tried to use the local legumes, but also to supply the biomass of the legumes as well with microalgae just to provide essential amino acids. Because also in the vegan dietary, for example, in the plant-based food, they're lacking the essential amino acids with microalgae have. Another aspect of projects that we are also covering and research area is feed and cosmetics. And this is another Horizon project, which is called Gene Beacon, where we are working on microalgae and gene editing in microalgae. So the idea is to have a wild type of strain of a microalgae, which is selected based on the lutein production, to upscale the cultivation of this strain, and then with residual biomass to try the poultry feed trials and extract the lutein. And then the other part is about gene editing and improving the strain or overproducing lutein, which is also helping to reduce the price. And also lutein, it can have application in a wide range of industries. And also through this project, we build a GMO facility where we can cultivate GMO microalgae now in the lab indoors. And it was certificated recently. We're working also on fuel. The project is called AlgaeSol, and this is a project where we are using different side streams, including also sun energy and CO2 to produce biofuel. And in this project, DTI has a role of contamination control strategies. So we want to develop methods to control the contamination of the microalgae cultivation for the production of biofuels. And this is an important step how to minimize the contamination and also keep the contaminants under control. And when I'm talking about contamination here, it's everything actually that is feeding on the microalgae, some protozooplankton or some ciliates. So they're doing grazing, they're eating the microalgae, they're reducing the productivity of the system. So we're looking how to keep it under control and also how to detect at the early stage the contamination. So I gave an overview of few actually project happening on Earth, and now there is a project happening in space next year, and this is a project together with Sophie Bionutrients and Uri Gravity, where they developed a science shell. So this is a small shell where we will cultivate chlorella in the heterotrophic way. So it will be without light, it will be closed in the dark box, and this really small box will be sent to space, to the International Space Station, because we want to see what's the effect of the microgravity environment, how is affecting the microalgae. And it's not only the microgravity environment, but also the cosmic radiation. But for the project, it's important this microgravity because we want to see what happens with the cell wall. But it's also, I think, relevant also for future projects because this can be food for the astronauts in the future. And this is one more application of microalgae. This is a recent project which is called Microfoam, and it's bio-based fire retardants using microalgal foam. So there is a species of microalgae called Phaeocystis, and they're creating big blooms in the environment, in the sea. Sometimes you can see on the coast a foam like this. So together with some partners, we will try to use a new advanced lighting system and also use some wastewater from the aquaculture facility to produce a big amount of this biomass, and also test if you can use this foam for fire retardants. Because usually the fire retardants used by now, they're like toxic and not good for the environment, and this can be something more bio-based and more environmental friendly than the previous one. And then some of the future directions, which I think maybe can be relevant for this network as well, because we are looking at different application of microalgae in different areas. And also one of the areas where I'm personally interested is to use the microalgae and try to connect the microalgae production and cultivated meat. And because we can use microalgae as a source of lipids and proteins, which can be also used as a component in the cultivated meat media, maybe some also growth factors, because there are some studies showing that some liquid fractions of the microalgae can improve the growth of some animal cells for the scaffolds as well. And also one of my biggest interests is these antimicrobial peptides from microalgae. So instead of using antibiotics, we can use antimicrobial peptides, and in this way also maybe try to reduce the use of antibiotics, which are creating this antimicrobial resistant bacteria, and also in the end testing the growth of microalgae on the spent media of the cultivated meat, because it can be rich in ammonium, which some of the microalgae like, and then we can also kind of close the loop. And this was the last slide. So thank you for your attention.
Thank you, Nicola. The floor is open for any questions from the audience. Is there any questions, or I can start? Eh? You have 2 claps. So maybe I'll start first. I'm because I'm a nutritionist, I'm a bit curious on why would microalgae be ideal for lutein production?
Because they're rich in lutein production.
Right.
Yeah, and also because it's more sustainable to produce microalgae, because you can also use other side stream to cultivate microalgae and then
Right.
extract the lutein. And I forgot to mention that we're also looking at sustainable ways of extracting the lutein from the microalgae as well.
Ah, okay.
Yeah.
But so besides lutein, there will be, I would assume, other types of bioactives that would be of interest.
Yeah, definitely. Because the idea at the beginning, I can say it now, it was a mycosporin-like amino acids, because microalgae are producing them to protect the cells against the UV when they're outside in the environment. But then all the strains that we were using in this project, they were not producing those components, and then we changed to lutein.
Okay.
Yeah.
Because those are a bit more high value products.
Yeah,
Suppose.
exactly.
Yeah.
Yeah.
Right. Are there other potential things that microalgae is useful to to use as a reactor, or you think that it can be, we can CRISPR things that we want into this? Yeah.
Yes, it's not only
Yeah.
for lutein production, but it can be the whole spectrum of compounds that we can look. We can also look at some bioactive compounds which can have application in the pharmaceutical industry and also in the biomedical
Right.
industry. Like
Right.
some drugs against cancer, for example, can be also isolated from microalgae, and then through CRISPR we can improve the overproduction of those components.
But then it also depends on the extraction efficiency and all this, right? Because
Yes, we need to have a good extraction efficiency, and also it has to be sustainable.
Right, right, yeah. Any questions from the audience? Otherwise, we can continue the discussion maybe after Alcine has finished her presentation as well.
I have a quick question.
Yeah.
This is more of a big picture question, but I've been to a lot of talks. Actually, was at one yesterday, talking about the problem of algae blooms in the environment, which as you know, occur downstream of any place where conventional agriculture is happening. So I know you're focused obviously on intentionally cultivated algae and possibly even engineered algae for specific purpose. But do you envision a future where maybe in the process of cleaning up the algae blooms we don't want, we could also harvest useful biomaterials from that?
Yeah, I was doing my PhD actually in harmful algal blooms actually, and those were the algae that are causing those blooms. And this is always kind of my idea. Okay, they are harmful for the environment, but maybe we can use them for something else. But the thing is that most of these algae, they're toxic. So it's actually, they're producing toxins. And then where could you use, for example, neurotoxins? Maybe they can have some function as drugs in the future, but yeah.
Okay, thanks.
For example, this what I was showing with the foam, they're creating a lot of problems for the environment and also for the aquaculture because they're blooming, creating the foam, so you cannot use the water on the seaside for anything else. For sport, you cannot use it, but also for aquaculture. And also when the biomass is sinking on the bottom, they're creating hypoxia and anoxia even, so they're making the whole ecological damage actually. But in this project, we're trying to show the good side of those microalgae as well.
Okay. Thank you, Nicola. Let's invite round of applause for Nicola first, and let's invite
Thank you.
Alcine over to let us to share with us her research. Alcine, are you able to share your screen? Yeah. And then maybe we'll continue the conversation. Yeah. Alcine is one of our postdocs under the department, and she will introduce herself better after I've get her to after I introduce her in a very a limited way. So Alcine, please introduce yourself to do yourself justice in terms of your experience in microalgae. Yeah.
Hello,
A
everyone.
better introduction to our department as well.
Okay, yes. I'll try to promote FST. Yes, I'm Alcine. I am a research fellow at NUS FST, and I have been with FST since undergrad. So I think total, including undergrad, PhD, and postdoc, coming to 11 years now. So so in undergrad, my research was mainly trying to cultivate probiotics in beers. And beer is a very hard media to grow probiotics because of the hops. So we managed to let it grow and survive for pretty long periods of time. And then for my PhD, I continued with that train of thought and then tried to cultivate probiotics in coffee. And coffee is also not easy to grow things because it's so nutrient scarce, barely anything can grow in it. But we managed to let the probiotics grow and survive for very long. And going along with the same train of thought, we then decided to grow microalgae, this time in side streams. And again, side streams, not easy to grow. Microalgae especially because for biomass purposes, we want a high amount of biomass, as high as possible. Otherwise, it's not going to be economically feasible. So throughout my research, it's mainly trying to adapt the substrates, adapt cultivation conditions, tweaking fermentation protocols to grow microorganisms in hard to cultivate media. Yeah, and throughout FST has been very supportive. So if anyone wants to join us, please, I advocate. Is that good enough, Dr. Liu?
Thank you, thank you.
Yes. So yeah, really FST has supported me throughout my research career so far. So yeah, just starting on my presentation today. So basically, I'm going to present what I did. Me and my PhD student Vivian have been doing for the past almost 3 years now. So for the past 3 years, we've been trying to cultivate microalgae in food side streams. And yeah, but before I begin, I just want to thank everyone here because we have 3 different time zones. And for people in Singapore here, we would have just finished eating dinner. And one of local typical dish that a Singaporean would eat would be cai fan. So what cai fan really means is economy rice. So it is a dish composed of rice and some meat, some vegetables, and maybe some tofu and egg. But for us in Singapore, we are presented with a very harsh reality because one day in a possible dystopian future, 90% of our food could disappear. And this may sound very dystopian, but if we are not careful, it could be a reality because Singapore imports 90% of our nutritional needs. And this makes us very susceptible to climate change, to pandemics, to geopolitical tensions. So because of this, the Singapore government is aiming to increase our local production from 10% to 30% by 2030, which is very, very ambitious because if you take things into perspective, Singapore is 195 times smaller than North Carolina. And we are not only 195 times smaller, but we are 10 times more population dense. And on top of that, within this small land mass, only 1% is devoted for agriculture. So you can imagine how hard is it for us to increase our local production from 10% to 30%. So for Singapore, we really need to start looking into farming land efficient food items. And one of the land efficient food source could be microalgae, because compared to conventional crops, microalgae is able to accumulate large amounts of proteins, lipids, and carbohydrates within a short span of time. And in particular, if we are growing microalgae through heterotrophy, which is the use of organic nutrients, then their productivity is way, way, way higher than what you would obtain with phototrophy or autotrophy, which is through photosynthesis. However, the caveat of using heterotrophy is that it relies on organic nutrients, meaning it relies on sugar crops. So this paradoxically actually reduces land use efficiency because we will need arable land to grow things such as sugar cane or sugar beets. And in addition, the sugar crops are also typically very expensive and has often been cited as the main economic hurdle in cultivating heterotrophic microalgae. So what Singapore could do to overcome this limitation with heterotrophy is to look towards our food side streams or food waste. So last year in 2023, we produced almost 800 tons of food waste and only 18% was recycled. So out of this 18%, the recycled food waste were things like soy whey, which is a byproduct of tofu production, and brewer spent grain, which is a byproduct of beer production. So in Singapore, what it means by recycled, it could be things like simply using them as low-value animal feed. But it could be, these sections could be better utilized for more higher value applications to grow microalgae. And at the same time, we're not only able to reduce food waste, but we might also reduce the reliance on sugar crops as well as replace synthetic mineral media. So because of this potential advantages, it is thus not surprising that others have also attempted to use these two same sex streams to grow microalgae, but very often their efforts did not produce enough biomass that could be economically feasible. So for example, for soy whey, it contains low to moderate quantities of monosaccharides, and very often the researchers required to supplement the soy whey with extra amounts of glucose to further increase the microalgae biomass produced. For brewer span grain, it potentially contains higher amounts of sugars, typically starch, trapped within the lignocellulosic material. But you will need to properly hydrolyze them to fully recover these nutrients. And if you're not able to properly hydrolyze them, then you will need to dilute them or add exogenous nutrients. In this case, the researchers diluted it with conventional media to grow euglena. So we wanted to differentiate ourselves from previous attempts. So we set ourselves with two aims. Number one, to, as much as possible, reduce the reliance on exogenous nutrients like glucose and conventional media. And number two, ensure that the biomass produced is high enough to be economically feasible, preferably more than 10 grams per liter. So with that, we set up on a multi-step approach. So this is actually our secret formula, which we only recently published. Yeah, so you may want to listen very carefully. Yeah, so in the first step, we optimize the hydrolysis protocols for each of the side stream. For soy whey, it has moderate amounts of sucrose. So this sucrose could be easily released to glucose and fructose with mild hydrolytic conditions. And that's what we did here. So 0.1 m HCl, 95 degrees, and 30 minutes was chosen after experimenting with some of the variables there. For brewer span grain, like what I said, it contains higher amounts of glucose, mainly in the form of starch. But because the starch is trapped within the lignocellulosic material, we had to implement a harsher hydrolysis protocol. So using response surface methodology, we derived the optimum hydrolysis condition that could recover 54 grams per liter of glucose. Now, because there's a harsher hydrolysis implemented for brewer span grain, you will notice that the amount of inhibitory compounds present in the brewer span grain hydrolysate is much higher than that in the soy whey hydrolysate. So the inhibitory compounds could be things like furans, could be acetic acid that are produced as a result of the harsh hydrolysis conditions. And if these compounds are present in excessive amounts, it could actually inhibit the microalgae growth, and then it would result in reduced biomass yields. So because of this, we then took the next step, which was to blend these two sex streams together. So the brewer span grain primarily was used as a nutrient-rich source containing high amounts of glucose, and soy whey was used as a diluent to reduce the levels of inhibitory compounds present in the final hydrolytic media. So we optimized this ratio through a simplex lattice mixture design, and after optimization, we derived a mixed hydrolytic media containing around 50 grams per liter glucose. And this actually supported a very high biomass of 22 grams per liter of oxynochlorella prototicoides. So through this multi-step approach, we have effectively achieved our two aims. Number one, high biomass. And number two, we completely replaced any exogenous nutrients, be it glucose or conventional media, in our hydrolytic media formulation. So when we compare our hydrolytic media to the standard medium, we find that the growth performance was comparable in terms of the growth curve, the biomass produced, the productivity, as well as the sugar to biomass ratio. And the reason why oxynochlorella could grow so well in the mixed hydrolytic medium was because it contained very high amounts of endogenous nutrients, such as glucose, phosphorus, and small amounts of extra nutrients, such as fructose, acetate, as well as ammonium and some amino acids as well. We also compared the economic cost of producing the standard medium to our hydrolyzed medium. And because we managed to completely reduce the amount of exogenous nutrients, the substrate cost was reduced by 96%. So raw material cost from $4.16 to just 15 cents. But because we need to hydrolyze these side streams to recover the nutrients, this eroded our cost savings, resulting in a total production cost of $2.76 to $6.40 per kg dry biomass. Now, right now, these estimates are not based on a techno-economic analysis, but these are only preliminary. But because the price ranges are comparable to that of BBM, then at least there is economic potential there. So we believe that the hydrolyzed medium could also be generalizable to other processes. So for example, if we want to cultivate other microorganisms that are more highly susceptible to the inhibitory compounds present in the brewer's bank grain, then we could simply increase the proportion of soy whey such that the amounts of inhibitory compounds are reduced. In another example, we could also boost the existing lipid and protein content in oxynochlorella. So for example, with the existing formulation of 15% soy whey and 85% brewer's bank grain, we can boost the protein content of 15% to maybe 30 or 40% by supplementing the hydrolyzed medium with other nitrogenous sources or other nitrogenous side streams such as chicken carcass hydrolysate. So this is exactly what me and my PhD student Vivian did. So in these graphs here, right, we have the hydrolyzed medium, and it was supplemented with equivalent amounts of nitrogen, mainly in the form of yeast extract, chicken carcass hydrolysate, arginine, and ammonium carbonate. And all nitrogen supplementations managed to boost the biomass protein content pretty well. But the most prominent effect was for the yeast extract. So yeast extract had the highest protein yield, primarily because of the greatest biomass achieved by day 5. So taking the yeast extract as an example, we then compared the hydrolyzed media to that of the standard media again, just to verify its effectiveness as a basal medium compared to the standard medium. And we found that the protein yield obtained in the hydrolyzed medium was higher than that of the standard medium. So on the graph on the right, the protein yield obtained was 7.2 grams per liter with the hydrolyzed and only 6.37 grams per liter with the standard medium. As a protein source, the oxynochlorella biomass grown in the hydrolyzed medium was also rich in essential amino acids. So for example, the first column is the essential amino acid composition of our oxynochlorella grown in the hydrolyzed medium. And if we compare the composition to that of the WHO requirement, which is the last column, it actually exceeded all the requirements for each essential amino acid. And the total essential amino acid content seems to be higher than other commercial oxynochlorella protodicroides out there, while also having comparable levels to that of other plant proteins such as soy or pea proteins. So ultimately, what we want for this project is to boost our Singapore's local food security, such that we have enough Cai Fan or economy rice to eat every day. And this is only achievable for Singapore especially, through a circular bioeconomy framework of utilizing our existing resources, such as soy whey and brewer's bank grain. But we need to be very sure that we implement the appropriate or optimum hydrolysis protocols such that we extract or recover the maximum amount of nutrients possible, such that the amount of biomass produced is maximized. So ultimately, the biomass produced will be used to produce seafood or meat analogs that could replace other land-intensive proteins such as beef or pork. So before I end off my presentation, I would just like to thank our funding source, National Research Foundation Singapore, and our collaborators at ETH Zurich and Singapore ETH Center. So if anyone is interested in more details that I presented today, please feel free to scan the QR code and it will take you to Bioresource Technology where more data is published. Thank you.
Thank you, Alcine. So any questions from the audience?
Hi, Alcine. Yeah, I just have just want to say thanks for a very comprehensive review of your research. I just have a very quick question on some of the inhibitory compounds that might have been produced from the hydrolysis. So you mentioned that furfural, HMF are some of the inhibitory compounds that you measured. How about other kinds of pentoses or any other kind of sugars? Would those also be inhibitory compounds, or would that affect maybe the growth of the microalgae?
I wouldn't say pentoses are inhibitory. In excessive amounts, like let's say if glucose is also present, pentoses in excessive amounts, I would think wouldn't affect unless it affects the osmotic pressure. But pentoses, I think instead of being inhibitory, are more non-preferred compounds. So the microalgae can still grow if they can if there are other assimilable sugars like glucose present. But if it's just pentoses present, then they are not able to assimilate them, then I don't think they will grow. So in this sense, there are more non-preferred compounds than inhibitory compounds.
I see. And this species is able to assimilate like pentoses, or fructose as well?
Yes, so this species can assimilate fructose. We also found that it can assimilate xylose and arabinose, but it was only assimilated after glucose and fructose was depleted. And when it assimilated these pentoses, for some reason, there was no increase in biomass observed.
I see.
Yeah.
Okay. Yep, thanks.
We have Matt.
Yeah, hi. Thanks for a nice talk. I was just curious about, did you somehow sterilize the substrates before the cultivation, or did you just cultivate after mixing the components?
Yeah. So in this case, just for experimental purposes, we filter sterilized it. Yes, but actually, if we were to scale up, filter sterilizing is not going to be feasible. So we think that actually because the filter sterilizing may be eliminated for upscaling, mainly because the mild acid hydrolysis regimes implemented here, so HCl and H2SO4, they actually enable us to maintain microbial sterility. So after hydrolysis, we actually plated it. We actually neutralized in the laminar flow and plated it, and it was sterile. So if you're going to do upscaling and you can actually invent certain processes that, like for example, pipings that could maintain sterility after hydrolysis, then you could actually do away with expensive autoclaving or expensive filter sterilizing. Yeah.
Okay, and then we have Nikola.
Yeah, I have also two questions actually regarding the side streams. So,
Yeah.
and also the inhibitory compounds. And do you know what is the phenolic phenol content actually of the side streams? Because sometimes those phenols, they can have an inhibiting growth on the microalgae as well.
Yes, so we didn't quantify individual phenolic compounds. So initially we only quantified total phenolic content based on the assay. And it was about, I can't remember the value, I think it was 2 point something gallic acid equivalent, mg gallic acid equivalents. But subsequently, we didn't quantify the phenolic compounds because we actually tested the growth of Dr Nikola just in the Brewers Penguin hydrolysis alone, and it could already grow. So we think that the inhibitory effect from these phenolic compounds is not as prominent. Yeah.
Okay,
Yeah.
thank you.
We also have Yeti.
Yeah, thank you for a nice presentation, both of you really. I was wondering that when there may be an option, and you definitely also said that's Nicholas on using media for cultivated cells like muscle cells. Do you have an idea of the impact of the anti-nutritional compounds, of which they would be, or would that be able to get by with a cut-off filter, or what nature does this have? You know, if we were going to use microalgae for that. So I don't know. I mean, Nicholas, you could also answer if you have an idea. So
sorry, can you repeat the question?
If we were going to use the microalgae as substrate for cultivated meat like muscle cells, the impact of the anti-nutritional components, as also said in this presentation, that there is an anti-nutritional component. So do we have an idea of what they are, what nature they've got, or if it's possible to sort of get rid of them by filtration or somehow?
Yeah, probably there are some downstream processing where you can get rid of those components. And also it's true that there are so many components that we don't know actually what their functions are. Because when I was working with toxic microalgae, I knew it, okay, this is the toxins, but then you have some other compounds which can, together with the toxins, they're kind of all having the same effect at the same time together. But if you remove one of those chemicals, it's a different effect actually. And this is also why we're always focusing on lipids and proteins, because we know how to actually, during the downstream process, how to get rid of the other components which are not nutritional for the microalgae.
Okay, thanks.
Alcine, do you have anything you could add in terms of applying the same concepts on in cultivated meat? Although I know that you're not working on cultivated meat.
So, like, I think you could remove through downstream, but the thing is, with more downstream processing, you're going to add more cost. So ultimately, we want the simplest way possible to cultivate it. So if there's already, let's say there's already inhibitory compounds present in your media, then you would try to find the simplest way to mitigate that. So for example, diluting with another media or using another cell line that is more resistant. So I mean, downstream, if it's easy enough, then by all means, but I would think that the simplest way is the way to go.
So you're
Yeah.
also saying that maybe also select for certain cell lines that
Yeah.
can grow within this condition, right?
Yeah.
Yeah.
Thank you.
Okay.
Maybe
So
it can be also done, if I can just add this, it can
of
be
course.
done both ways, maybe to improve the microalgae, like using gene editing tools to produce more of these compounds, and then also having the cell line that is more resistant to something that is actually coming from the microalgae that might inhibit the growth of the cell line.
Yep. Actually, this question is a good way to end today's session as well, because the upcoming research, the upcoming seminar, online seminar for next month would be on cultivated meat and so on. I hope we can pull that off. So, but please look out for that particular announcement. It will happen next month. So we will try to have a few of these seminars at least in the next few months on a regular monthly basis, so that different groups of people can share what they are doing and then have some exchanges of ideas and so on. Yeah. And Professor Young is also the lady that sent the, what is it? It's a fellowship for people who are interested. So I've already forwarded to many of the Singapore, the people that are in Singapore. Hopefully they can apply for this particular fellowship that was circulated around for the advanced studies. Yeah. Okay, Louis, do you want Louis and Rohan? I forgot to introduce Rohan, who is Rohan. Where are you? Are you still
Yes,
here?
I'm still here.
Do you want to introduce yourself a bit, because you are the co-chair, and I'll pass the next seminar to you.
Yeah, no, I just I'm
Yeah.
Rohan Shirvaikar. I'm at NC State. I'm also a professor here and a co-director of the Bezos Center here. And I want to thank both our speakers today for the fantastic seminars. And yeah, as we go through these monthly seminars, we are going to try to tackle new and diverse topics. So the next one will be on around cultivated meat, and then we'll target some of the other topics this whole group has identified as high priority and of interest in the coming months. And Luis, I just wanted to remind everybody of the email you had sent to mark your calendars for the visit to the US for the ICAN delegation in January. Tomorrow, in fact, some of us are meeting to start planning that visit, so more details will be forthcoming. But we're looking forward to welcoming you in North Carolina first and then in Boston.
Okay, Louis, would you like to just close the session?
Of course, yeah, I'm happy to. Yeah, I want to thank you very much, both of our speakers. I think it was really interesting. I think the range of different projects and applications where microalgae can provide solutions is extremely fascinating. And I thought, yeah, you gave some excellent presentations today. I want to encourage that you reach out to me or Rohan or maybe if you are interested in getting in contact with our speakers today, and we're very happy to facilitate contact. And I also want to say that the recording of today's meeting will become available. So if you have some colleagues who were not able to participate today, or and you would like to share this webinar, we will make that an option. So please keep that in mind. But apart from that, I will say thank you. Have a great night or afternoon or day, depending on where you are in the world. And we'll see you next month for an interesting webinar, or at least some of you, for an interesting webinar on cultivated meat. Bye, everyone.
Okay, bye, everyone.
Thank you. Bye.
You.
Bye.
Bye. Bye. Thanks, Nicola. Thanks, Alcine. Thank you.
Thank you so
Okay.
much, Mei Wei. I thank you for for someone who's sitting there late at